Have you ever heard of gram staining? This video explains the principle behind gram staining and also tells you how it is done. By now, we all know that most of the bacteria have cell walls in them. On the basis of the composition and structure of their cell walls, bacteria can be distinguished into gram-positive and gram-negative bacterial cells. But what do we exactly mean when we say gram? The term gram comes from the name of a Danish bacteriologist named Hans Christian Graham, who, while studying pneumonia causing organisms in 1992, developed the method of gram staining. As we have discussed before, gram staining is a method of staining used to classify bacterial species into two distinct groups. Gram-positive bacteria, the colonies of which are stained purple or violet at the end of the gram staining procedure, or the gram-negative bacteria whose colonies are stained pink at the end of the gram staining procedure. Now let's move on to how we can perform a gram staining procedure. The first step is the preparation and fixation of a smear onto a slide. The first step begins with applying a smear of the test bacteria onto a clean glass slide. This is followed by air drying and heat fixing the smear by passing it through a flame a few times. Step number 2 begins with flooding the slide with crystal violet stain and letting it stay for 1 minute. The excess stain is washed off by rinsing the slide with water. In the next step, grams iodine is used to flood the slide. Grams iodine is allowed to stay on the slide for 1 minute. Excess stain is washed off by rinsing the slide with water again. Step number 4 is the use of a decolorizer. The slide is this time flooded with a decolorizer, which may be alcohol or acetone or a mixture of the two. This is allowed to stay on the slide for 5 up to 15 seconds, and then the slide is again rinsed with water. In the step 5, the slide is flooded with safranin, which is a counter stain, and it is again left for 1 minute. The excess stain is washed off again by rinsing the slide with water. The excess water is blotted off using blotting paper, and the slide is air dried. Finally, the slide is observed under a 100x microscope using oil immersion. Moving on to the principle of the gram staining procedure. The principle of gram staining procedure is the ability of the bacterial cell walls to retain the crystal violet dye, which actually depends upon its chemical composition. Now, why do we only use crystal violet, grams iodine, decolorizer, and safranin? Crystal violet is the primary stain. Initially, all the bacterial cells take up the primary dye, which is the crystal violet. The cells that have the ability to retain this dye are called gram-positive cells. However, cells that lose the lipid layer in their cell walls also lose the ability to retain this dye, which is why these cells are called gram-negative bacterial cells. Note that gram-positive cell walls have a higher lipid content and gram-positive cell walls have a higher peptidoglycan content, which is why, as the lipid content of the gram-negative cell walls is lost, they lose the ability to retain the primary dye. Grams iodine is required for the formation of crystal violet iodine complexes to prevent the easy removal of the crystal violet dye from the gram-positive cell walls. The decolorizer or the solvent helps remove the lipid layer from the cell wall, which in turn means that the primary dye gets removed from the gram-negative cell walls. In contrast, in the gram-positive cell walls, the decolorizer closes the pores by dehydrating the cell walls, which makes it hard for the cell wall to lose the crystal violet iodine complexes. The safranin is the secondary dye or the counter stain. It gives color to the gram-negative cell walls. As the dye is lighter and weakly water soluble, it can bind to the gram-negative cell walls but not to the crystal violet-bound gram-positive cell walls. Well, that was all about gram staining. If you have any queries, don't hesitate to ask for our help. Thank you.